Hi ladies and gentlemen, I am Muneeza Fatima and I am presenting a project known as Implementing Pleurotus osteitis mushrooms for nitrogen mitigation and a way to restore salt marshes. According to the previous research of Alongi 2020 and Barbier 2011, salt marshes are important ecosystems since they provide various services such as climate stability, carbon sequestration, water filtration, and denitrification that you see in the right figure. However, our research that has been proposed by Perez Rizafa 2019 aware us that these ecosystems are degrading over time by inputting excessive reactive nitrogen into the environment. Additionally, previous studies from Deegan 2012 emphasized our marsh loss and made it clear that excessive nitrogen causes eutrophication problem. And if you see in the left figure, eutrophication decreases below ground biomass and increases above ground biomass of vegetation, such as Spartina alterniflora. These evolutions in the ecosystem decreases the ability of salt marshes to protect the environment against storm surges. Therefore, oyster mushrooms are being used, and according to Agandal 2014, Salzberg 2015, Miles and Chang 2004 studies, oyster mushrooms require nitrogen between 1.84 to 2.08% range in their cultivation process. So the nitrogen that is in excess in this environment would be useful for mushrooms development. To investigate the health of marsh and find the average rate of marsh loss, three different marshes of Long Island, such as Crab Meadow, Sunken Meadow, and Timber Point, were monitored. The red circle on these marshes show the locations that were studied in this paper. The width distance from three different marshes and three different regions, including opening point, middle point, and ending point of mosquito ditches, were measured in meters over 22 years on Google Earth Pro. The data was collected in meters and analyzed on jump variant 16. For the development of oyster mushrooms, two experiments were conducted. One was set up in green spawn substrate and in 0 mg per liter, 5 mg per liter, and 15 mg per liter sodium nitrate concentration. However, for the second experiment, an agar as a substrate was utilized, and 0 ppm, 5 ppm, 15 ppm, and 18 ppm sodium nitrate concentrations were used. Grain spawn experiment was sterilized in autoclave. On the other hand, sterilization process was not applied in agar substrate experiment. Both experiments were conducted in greenhouse for 1.5 months, and once mushrooms cultivation was completed, fruiting bodies of mushrooms that were grown in grain spawn were weighed on electric balancer. Although the diameter of mycelium in agar substrate was measured with a ruler every day. The data of both experiments were obtained and analyzed on jump variant 14, and Tukey test was used to compare means of these experiments. For the results, the data that was collected from mosquito ditch measurement of Crab Meadow Marsh was analyzed and graph was built with standard errors on jump that you can see in the left figure. The results show that there is no significant difference between pairs over 22 years. All HSE threshold matrix values were negative and p-value was greater than 0.05. The average growth of opening point of Crab Meadow is 1.9 meters for every 11 years. Additionally, Sunken Meadow data sheets were also analyzed on jump, and according to the negative threshold matrix value, there is no significant difference between pairs that you can see in the right figure. The average growth of opening point on Sunken Meadow Marsh is 0.73 meters. Furthermore, for the timber point, there was also no significant difference between pairs due to the negative values of HSE threshold matrix. These three marshes are also connected by the same letter A, which indicates that there is no significant difference between pairs. Moreover, the average growth of opening point of Timber Point mosquito ditch is 0.57 meter for every 11 years. Unlike Crab Meadow, Timber Point and Sunken Meadow has less growth rate. Consequently, the present study from Gobbler 2017 and Crossby 2016 confirmed that Crab Meadow is turning into mud flats more frequently, since it has been influencing by eutrophication. It is understandable that compared to Timber Point and Sunken Meadow, Crab Meadow is the only marsh which is losing rapidly due to the highly dense areas near Crab Meadow. Moving further, when sites of these marshes were compared to each other by ANOVA, the results demonstrated that there is a significant difference between Crab Meadow and Timber Point pairs in all years, such as 2001, 2011, and 2022. Figures also show that scrap meadow and sunken meadow pair has also a significant difference. The significant difference of these sites were found by the positive value of HST threshold matrix, and p-value was very less than 0.0001. Now the data that was collected by mushrooms growth was also analyzed on jump variant 14, and the results demonstrated that not only control and low concentration of nitrogen favors the mushroom cultivation, but also higher concentration of nitrogen, which is 15 mg per liter in grain spawn substrate and 18 ppm in agar substrate, responded positively and increases the growth rate of oyster mushrooms and decreases the production time. Thus 2011 research supports my results that nitrogen enhances mushroom biological efficiency and increases growth rate. If we compare both graphs, it was found that species in agar substrate in 15 ppm concentration has less growth than compared to grain spawn substrate with 15 mg per liter concentration. Therefore, according to Agundal 2014 studies, it has been concluded that substrates also affect the development of fruiting bodies of mushrooms. For future works, I will be continuously working on aerial imaging by Google Earth Pro to find the health of global marshes over time and will collect data for other students to make them easy. Instead of using reactive nitrogen for growth, I will use vegetation rock and other dead organic matter as a fertilizer to mitigate eutrophication. I will work with other mushrooms as well to find the better salt and nitrogen tolerance mushrooms that might be more useful and beneficial in restoring marshes and decrease the growth rate of mosquito ditch. Thank you.